Okay, right. I'm going to do one more practice problem involving fitness, and then we'll switch over and talk about inbreeding for a little bit. So in your practice problem sheet, if you can turn to practice problem 6, this has to do with an experiment carried out on a laboratory population of the fruit fly, your friend and mine, Drosophila melanogaster. This one's interesting because it involves a gene that has an allele that's an embryonic lethal. The allele for bristle shape, or the gene for bristle shape, comes in two alleles, stubble and wild type. Stubble looks like, well, stubble. Wild type has nice long bristles. Stubble is actually dominant to wild type. You probably know this, but just as a reminder, wild type means, the average phenotype that you'd find in a wild population. It's, well, normal in a sense, but it's not necessarily dominant. My 1400 students sometimes get mixed up with this. You can have alleles that are normal and common, but they're also recessive. And stubble is dominant to wild type, with the twist that flies that are homozygous for stubble always die during embryonic development. That's an embryonic lethal. Okay, so how do we work this? Shepherd starts with 86 normal flies and 86% normal flies and 14% stubble flies. Let's calculate the allele frequencies. So here we go to the whiteboard. So we're starting with 86% wild type flies. And 14% stubble flies. All right. Now, wild type flies have the genotype. Let's do stubble with a capital S and non-stubble with a lowercase s. So we know this genotype is the heterozygote, big S, little s. Whoops, no, we don't. The wild type flies are homozygous recessive, little s, little s. Stubble flies are big S, little s. And we don't have any of these big S flies, because that's an embryonic lethal. Eggs, fertilized eggs that have that genotype never develop into adult flies for some reason. Okay, so what is our allele frequency? Okay, simplest way to do it is frequency of, let's say, the little s allele is equal to the frequency of the homozygotes plus 1/2 times the frequency of heterozygotes, Wagner. And so that's going to be equal to 0.93. If you want to do it the long way, I don't really care how you do these problems as long as you get the right answer, but you could imagine a population of 86 Wagner wild type flies that are homozygous recessive and 14 heterozygous flies that are stubbled. That's 100 flies, which means there's 200 alleles. And now the frequency of the little s allele is equal to, let's see, that's 86 homozygous flies, which have 2 alleles each. and 14 heterozygous flies, which have one little s allele each, all divided by 200 total alleles. So if you have a percentage problem like that, you can temporarily pretend that the percentages just convert into a population of 100 individuals. So we don't know the actual population size that Shepard was working with, but we can temporarily pretend it was exactly 100, and we can solve the problem that way. And if you do 86 times 2, that's what, 172 plus 14 is 186 divided by 200, and that's equal to 0.93. You get exactly the same answer. So if you want to remember a little extra formula, you can just say that the frequency of any given allele is equal to the frequency of the homozygotes plus half the frequency of the heterozygotes. If you don't feel like remembering that, you can just temporarily pretend that your percentages equals the number of individuals in a population of 100 and solve it like we've already seen. Any way you want it, that's the way you need it. Right. And then, of course, the frequency of the big S allele is just 1 minus the equal sign, 1 minus the frequency of the little s allele. That's going to be equal to 0.07. All right, so we can call that 0.07 P and that 0.93 Q. It actually doesn't matter whether we use P for dominant and Q for recessive or the other way around. It doesn't matter which one's dominant and which is recessive, or even if any of them are. I often assign in problems like this P to the dominant allele frequency and Q to the recessive allele frequency, but it's not important. You should get the same answer regardless. Okay, so switching over temporarily to the practice problems again. Assume for right now that the wild type and the stubble flies don't differ in fitness. How do we calculate the mean fitness? And then how do we predict the percentages of normal and stubble flies in the next generations? Okay. All right. How do we do that? Okay. Temporarily stop the share, I see. Just a second. Oh, there's the share screen. I think I'd know this shit by now. Okay, back on the whiteboard. Okay, so what we ultimately want to predict is what are the allele frequencies going to be in this population after one round. Okay, the probability of an SS fly dying before it gets to reproduce is some number. It does not matter what it is. I'm going to call it X. The probability of a big S little s stubble fly dying before it gets to reproduce is the same number, because we're assuming there's no difference in survival or reproductive ability between the stubble flies and the wild type flies. The probability of a big S fly dying without reproducing is equal to 1. I'm going to rethink that. Let's change that to the probability of a little s fly reproducing successfully. And that's some number that I'm going to call X. The probability of a heterozygote fly reproducing is also going to be X. And the probability of a homozygous dominant fly is equal to 0. right? No chance that a big S fly is going to reproduce. We can use that to calculate the relative fitnesses. We can divide all of those probabilities by the highest value among them. So the relative fitness, that's a W of the little s genotype, is equal to X divided by X, which is 1. The relative probability of a heterozygote fly reproducing successfully is X divided by X, which is 1. The relative probability of a homozygous dominant fly reproducing successfully is 0 divided by X, which is 0. No chance that a homozygous dominant fly is going to reproduce. Okay, with me there. What we now get to do is calculate relative fitness. And it goes a little something like this. Our equation here is p squared plus 2pq plus q squared equals W with a bar on it. There, W bar. Okay, we've got to add to that though. Sorry, trying to get this thing to insert where I want it to. Okay, we have to add something to that equation. If all of our fitnesses were equal to 1, then we get p squared plus 2pq plus q squared equals 1, which is just what you get when you square the expression p plus q equals 1. If you square both sides of that, you get p squared plus 2pq plus q squared equals 1. Here's the thing. We have to add these W terms to our equation to show the effect of selection. If all of our fitnesses were equal to 1, there wouldn't be any selection going on, just because there would be no difference in the probability that individuals of any genotype would get to reproduce successfully. Okay, so here we calculate W bar by going p squared times W big S. Okay, I'm going to go, all right, I'm going to, let's get rid of that. P squared times W big S plus 2pq W big S little s plus q squared W little s equals W bar. There's the bar. All right, how do we actually calculate that? Well, that's 0.07 squared times the fitness of the big S genotype, which is 0. Okay, that's pretty easy to do. Plus 2pq times 2 times 0.07 times 0.93 times 1. That's 2pq W big S little s plus 0.93 times 0.93 times 1. That's q squared times W little s, and that gives us W bar. Okay, so I need somebody to plug and chug what I've just done. 0.07 squared times 0, that's easy, plus 2 times 0.07 times 0.93 times 1, plus, sorry, I just realized that should be a plus, not an equal sign, plus 0.93 times 0.93 times 1. Plug that all in and you should get, anybody got that for me real quick?
It's 0.995.
Okay, 0.995. Okay, I see it now. Okay, thank you, Michelle. I just saw the chat. Is 0.995. Okay, that's W bar. That's our mean fitness for the population. Our predictions for what should happen after one generation our predicted frequency of big S is going to be, we get these predictions by taking that equation that I just wrote over here, by taking this and dividing all of its terms through by W bar. So that gets us P squared times W big S over W bar plus 2PQ times W big S little s over W bar plus Q squared W big S over W bar equals W bar over W bar, which is 1. So we take that and we have to divide all of its terms through by W bar. So the frequency of the big S genotype in the next generation is going to be equal to 0.07 times 0.07 times W big S, which is 0, divided by 0.995, which is W bar. And of course, that's going to be equal to 0. It doesn't matter what the other numbers are. 0 divided by anything is still going to be 0. Okay, so the frequency of big S homozygotes in the next generation is 0, because they're all dead, right? Because this is an embryonic lethal. No real surprises there. The frequency of the heterozygotes, on the other hand, the frequency of the stubble flies is going to be 2 times 0.07 times 0.93 times W big S little s, which is 1, divided by 0.995. Now think about this. This little term I put in parentheses here is 1 divided by something smaller than 1. That's going to be something greater than 1, right? 1 divided by something that's less than 1 is going to be equal to something that's greater than 1. 2 times 0.07 times 0.93, that is what you would expect that genotype frequency to be if there were no selection. That's just 2PQ. If we're in Hardy-Weinberg land, the frequency of that genotype in the next generation should just be 2PQ. 2PQ multiplied by the relative fitness divided by W bar, that relative fitness over W bar term, this right there, that is the number that gives you the strength of selection. If it's greater than 1, then the frequency of that genotype is going to be higher than it would be in the absence of selection. And if it's less than 1, that is, the genotype frequency will be less than what it would be if there wasn't any selection going on. So that quantity is equal to 2 times 0.07 times 0.93 times 1 over 0.995. If anybody feels like plugging and chugging, you can type it in the chat if you don't feel like talking. Drum roll, please. 0.13 or 0.131. Okay, 0.1, yeah, 0.13 is good. 0.131 if we want to go an extra digit. Okay, great. And finally, last but not least, our frequency of little s wild type individuals is going to be 0.93 times 0.93, which is what the frequency would be in the absence of selection, times 1 divided by 0.995, 1 divided by 0.995, and that 1 divided by 0.995 again tells you that's greater than 1. So the frequency of homozygous recessives with normal bristles is going to be higher than it would be if we did not have any selection going on. And so that number is equal to, hang on, seeing, okay, coming through on the chats, 0.87. Okay, right. I'm going to round it to 2 digits, but yeah, that number is going to be 0.87. Okay, so that's the second part. If Shepard took this population and just simply let it, just simply left it alone, then after 1 generation, we would expect to see the frequencies of genotypes go from 86% wild type and 14% stubble to 87% wild type and 13% stubble. Okay, not an enormous change, but certainly one we could measure, certainly one we could test experimentally. Okay, hope everybody is still with me. I'm going to go ahead and clear this. Actually, I could save it. Okay, I have actually saved this now. I'll post it if anybody wants me to, but I'll clear it. So let's clear all of this and let's go back and look at the practice problem. Shepard introduced an additional source of selection by removing 60% of the wild type flies before they could breed in each generation. So let's repeat part B, but take this into account, that in Shepard's original experiment, he'd take out 60% of the wild type flies before they could breed. Okay, so here again, we're starting with 86% little s wild type flies and 14% big S little s double flies and 0% big S flies because that's an embryonic lethal. Okay, the probability that a big S little s stubble fly, yeah, survives and reproduces successfully. Dang it, hang on. Sorry, I didn't mean to do all of that. Okay, 86% SS flies, 14% homozygous flies, stubble, and 0% homozygous dominant flies, because those are embryonic lethals. Okay, now the probability of a big S, little s fly reproducing successfully is some number, and it doesn't matter what it is, we can call it X. The probability of a big S fly reproducing successfully is equal to 0. What's the probability of a wild type fly reproducing successfully? Well, it's equal to, all right, it's equal to the probability of surviving and reproducing normally, which will set to being the same thing as the probability for big S, little s flies, right? We're calling it X because it's not a given that any fly successfully reproduces. Any given fly might not for any one of a number of reasons. But if left to their own devices, stubble flies and wild type flies, we're saying in this problem, have equal probabilities of surviving and reproducing. However, there's also the fact that the experimenter is removing 60% of the wild type flies before they can reproduce at all. So if you're a wild type fly, what's the probability of your reproducing successfully? It's the product of two separate probabilities. X is this mystery probability that you will, not die of disease or be too ugly for anybody to want to mate with you or whatever it is, multiplied by the probability that the experimenter is going to take you out of the experiment and feed you to the fish or whatever you do with excess fruit flies. And the probability of the experimenter doing that is 0.6, and the inverse of that is 0.4. So the probability of a big S or of a big S fly reproducing successfully is 0. The probability of a heterozygous stubble fly reproducing successfully is some number, that's X. The probability of a wild type fly successfully reproducing is the probability of successful reproduction if it doesn't get taken by the experimenter, multiplied by the probability that it doesn't get taken by the experimenter. So that's X times 0.4. I hope that came out somewhat clearly. Sometimes, if we're lecturing in person, I can see puzzled faces and go back and try to explain things again. Sometimes that makes it worse, but I can try. When I can't see faces, I'm not always sure how well I'm getting through. But forge on we must. This allows us to calculate our W's by dividing all of these by the largest value. So the relative fitness of the big S individuals is 0 divided by X, which is 0, duh. The relative fitness of the big S, little s heterozygous genotype is X divided by X, which is 1. And the relative fitness of the little s individuals, the wild type, is X times 0.4 divided by X, which happens to be 0.4. So what this is saying is that if you are a wild type fly, your chances of reproducing successfully are only 40% as good as the chances of you successfully reproducing if you are a stubble fly. So that's how you model the effect of selection here. And now we can take all of these and put them into our Hardy-Weinberg equilibrium. So what we have to do now is calculate W bar, and that's P squared times W big S plus 2PQ times W big S little s plus Q squared times W little s equals W bar. There's the bar. All right, now what? Let's go ahead and plug that in to calculate W bar. Well, we'd already established that P is equal to 0.07. It's the same, we're starting with the same values that we've already calculated. So 0.07 times 0.07 times 0 is equal to 0. Duh. Plus 2 times 0.07 times 0.93 times 1. That's 2PQ times the relative fitness of the big S little s genotype, plus 0.93 times 0.93 times 0.4. Plug all of that in, and what we get is W bar. All right, I'll pull up the chat if anybody wants to tell me what that is.
It should be 0.048.
0.048?
0.48, yeah.
0.48, okay. Anybody else get that? Sounds right to me. I'll go ahead and 0.48. Okay, great. There we go. So that's our relative, that's our mean fitness for the population. Now, to work out what should be the genotype frequencies in the next generation frequency of big S is going to be P squared times W big S divided by W bar. There's our W bar. There should be an equal sign in there. And now I can't go back and add it. There, close enough. There's an equal sign. And remember, we're getting this by taking this equation right up here and dividing all of the terms through by W bar. So we end up with P squared times W big S over W bar plus 2PQ times W big S little s over W bar plus Q squared W little s over W bar equals W bar over W bar, which happens to be 1. So that's where we're getting these. So our new frequency of big S individuals is going to be 0.07 times 0.07 times 0 divided by 0.48, which is equal to 0. Okay, that was easy enough to figure out. All right, our frequency of heterozygotes in the next generation, big S little s, is going to be equal to 2PQ times W big S little s, the relative fitness of that genotype, divided by W bar. There's the bar. And remember again that these terms right here, that tells you how strong the selection is. 2PQ is what the allele frequency, or the genotype frequency rather, would be in the next generation if there wasn't any selection happening. If there wasn't, we'd be back in Hardy-Weinbergville, but we're not. So this term, the relative fitness divided by the mean fitness, gives you the strength of selection that's going on. And that's going to be equal to 0.07 times 0.93 times 1 divided by 0.48. 1 divided by 0.48, that's close to 1 divided by 1/2, which is 2. So the frequency of heterozygotes in the next generation is going to be almost twice what it would be if there wasn't any selection going on. And Garen's already calculated that that's 0.28 if you plug and chug the numbers. All right, finally, I mean, from here, you could calculate the frequency of the wild types as just 1 minus 0.28, but let's go through it the long way. Frequency of little s homozygotes is Q squared times the relative fitness of that genotype divided by W bar. There's our bar. And that's going to be equal to 0.93 times 0.93 times 0.4 divided by 0.48. Now, 0.4 divided by 0.48 is 5/6. That's going to be about 0.83. So they're going to be about 5/6 as many homozygous recessive individuals in this population after one generation as there would be if there weren't any selection going on. So that is going to be equal to, anybody who wants to plug and chug that is welcome to. I'm going to say that's going to be about 0.72. Check me, am I right? Okay, Thomas says 0.72. Okay, there we go. We did it. That's the problem. So that's how you do that. I'll go ahead and save that if we need to refer to it again. Okay, in the time we've got remaining, I want to talk about inbreeding. This being Arkansas, you can insert your own jokes here if you particularly care to. Let's go ahead and find a practice problem that involves inbreeding. Let me try to find the one about 8's fun. Okay, 9 is a fun one. Problem 9 is about a species of wild oat called Avena fatua. This was a study of wild oat population genetics in California, and they looked at a trait called pubescence, which just means hairiness on the leaf sheath. And this is apparently controlled by a single locus with 2 alleles. And in this particular species, you can tell the heterozygotes apart from the homozygotes because apparently there's incomplete dominance going on. So leaf sheath hairiness is controlled by a single locus with 2 alleles, and the frequencies of genotypes in one population are big L, little l, and little l, little l. Okay, we'll start by calculating the allele frequencies. And let's go to the whiteboard. And clear the whiteboard. Okay, so in these wild oats, we have big L oats are at 57.1% of the population. Heterozygous white oats are 7.1% of the population. And homozygous recessive wild oats are 35.8% of the population. So that's a lot of wild oats. Okay, now what? First of all, we want to calculate the allele frequencies. The short way of doing it is we'll let the frequency of the big L allele, we'll call that P, and that's going to be equal to 0.571, the frequency of the homozygotes, plus 0.071 divided by 2, half the frequency of the heterozygotes. So that is going to be 0.571 plus 0.0355. That's going to be 0.6 something. It's not going to be 0.605, I think, but I'm going to round it to 0.6. Okay, I'm not seeing anything coming through on the chat, but I'm going to assume I'm right because I've got a PhD, so yeah. Okay, so the frequency of the little l allele we'll call Q, and that's, okay, 0.61. Okay, 0.61, close enough. That frequency is going to be 0.358 plus 0.071 divided by 2, and that's going to be equal to 0.39. If I wanted to do this the long way, I could say, let's pretend we have a population of 1,000 oats. 571 are big L, 71 are big L, and 358 are little l. Calculate the allele frequencies from that data. We could do that if we wanted to. We'd end up getting the same answer. Okay, so that's our allele frequencies. Watch this. If we were under Hardy-Weinberg, our expected frequencies of big L individuals would be 0.61 squared, which is actually going to be something like 0.37, 0.38. It's going to be almost 0.36. Anybody who wants to calculate that can go ahead and do so. What's 0.61 squared? 0.37. Okay. All right. If we were in Hardy-Weinberg, then the frequency of heterozygotes would be 2 times 0.61 times 0.39, which would be about, I should have just rounded off to 0.6 and 0.4 because I can do that in my head, which would be about 0.48. And happens to be 0.48. And then our frequency of little l's is going to be 0.39 squared, which is going to be, clickety, is going to be about 0.15. Okay, we got it. Okay, look at the difference. If we were under Hardy-Weinberg, we would expect to have a heck of a lot more heterozygotes than we do. We would expect that almost half the population would be heterozygotes. In the real population, less than 10% of the population is heterozygotic. This is the signature of a population that's undergoing a lot of inbreeding. Inbreeding by itself doesn't cause birth defects. Inbreeding per se is not the reason why, you have extra fingers or limbs in places they're not supposed to be, or a tendency to sit on the front porch and play the banjo and look around in a menacing way. What inbreeding does is boost homozygosity. Now, the reason why it's associated with defective offspring is this. In humans anyway, every one of us is carrying an average of 3 alleles that would cause really severe health problems if they were homozygous. You know, we're all genetic carriers of something, probably more than one thing. What makes it possible for the human race to persist and for many babies to actually be born quite healthy is the fact that most of us aren't carrying the same deleterious recessive alleles. My wife and I don't share any ancestry in common as far as we know, and I know most of mine back 5 generations, and I'm pretty sure that our ancestry doesn't overlap, at least for the period of time where we have historic records. So I might be carrying a recessive allele that would be very bad if it were homozygous. She might be carrying a recessive allele that would be very bad, but the odds that we're carrying the same recessive allele are very, very low. On the other hand, if I'm carrying a recessive allele and my sister is carrying one, the odds are much greater that it's the same recessive allele, because my sister and I, by definition, share a lot of genes in common because we had the same parents. So if I'm a carrier of, I don't know, cystic fibrosis, then the odds are high that my sister is also a carrier of cystic fibrosis, because I would have gotten it from one of my parents. She's got the same parents. There's a decent chance she could be carrying it as well. So fortunately, I think she's old enough that this is no longer an issue, never mind. Despite growing up in Louisiana, we've never been tempted that way. Yeah, but sorry. Even if, okay, now I just completely derailed my own train of thought because yuck. But if we were to have a kid, we'd have a relatively high chance of giving birth to a kid who got the same negative recessive allele from both of us. Whereas I had kids with somebody who is not closely related to me, and the odds are much lower that our son is carrying the same deleterious allele. Okay. How do we mathematize that? We calculate a quantity and we call it F. And F is equal to the probability that 2 alleles are identical by descent, which simply means that they are inherited from a recent common ancestor. Take 3 people. There's me, there's my sister, and there's the Emperor of Japan. All 3 of us have brown eyes. However, my sister and I both got our brown eyes from our father. To a very rough approximation, brown eyes are dominant to light eyes, and my mother has light eyes. Hers are green. So my dad has brown eyes. So if I have brown and my sister has brown, we must both have inherited that allele from my father. On the other hand, I don't have any common ancestors with the Emperor of Japan unless you were to go back probably something on the order of 10 to 20,000 years. I'm not sure. It doesn't really matter what that number is, but I have no Japanese ancestry. I certainly don't have any ancestors that were in the Japanese royal family. So whatever the alleles are that I have and that the emperor has that are making our eyes brown, they don't descend from a recent common ancestor. They may not even necessarily be the same allele. You know, my DNA and his DNA have probably independently undergone a number of mutations in the sequence that's coding for eye color. So, you know, they're not even necessarily identical alleles anymore, and they may not have been for a long time. So that's what I mean by alleles being identical by descent. We can calculate this as equal to 1 minus the observed over the expected heterozygosity. That capital H is heterozygosity, and all that is the frequency of heterozygotes in the population. So here, the observed heterozygosity is 7.1% or 0.071. Our expected heterozygosity is 0.48, right? 48%. So if the population is not undergoing any inbreeding, then homozygosity over heterozygosity is 1. 1 minus 1 is 0. F is going to be 0 in a population where there is no inbreeding. The more inbreeding is going on, then the smaller observed heterozygosity gets relative to expected heterozygosity, and the smaller that fraction gets, and the larger 1 minus that fraction gets. So to calculate that here, that's equal to 1 minus 0.071. That's the observed heterozygosity divided by 0.48. That's the expected heterozygosity. And let me see, 0.071 divided by, okay, Garen's calculated that, that's 0.85. That is a population that is rather highly inbred. In extreme cases of inbreeding, there might be no heterozygosity left at all. And there's actually a case where we've deliberately gone out and done this. There's a laboratory, the Jackson Laboratories, that breeds for biomedical research all over the world strains of inbred mice. If you do, we used to have a researcher here that was using them for some work. She has retired, so I don't think anybody at UCA is using these, but you'll encounter this if you go on to medical research. You'll encounter inbred mice. And inbred mice are exactly what that sounds like, marrying, not really marrying, but mating brothers and sisters for a minimum of 20 generations, and in some cases a lot more than that. Yeah, we've deliberately created mouse strains that are the product of brother-sister matings for 20 generations, or in some cases, hundreds of generations. Why the hell would you want to do that? Well, it's a lot cheaper than cloning. Inbred strains of mice are all homozygous at every locus. They're essentially genetically identical. There's almost no genetic variation left, because when you inbreed, what you do is jack up the frequency of your homozygotes. That makes them useful for certain experiments because a given strain of inbred mice, the individuals are genetically all the same. You no longer have genetic variation as a confounding variable. You can test a drug on a strain of inbred mice, and all of them should respond to it in the same way. There aren't going to be some that are genetically more resistant or more susceptible or what have you. The other reason you would want inbred mice is many of them are homozygous for a negative allele that mirrors a health condition in humans. So you can order inbred mice that are all guaranteed to be homozygous for a gene that's strongly linked with diabetes. These mice will all develop diabetes if you feed them the right diet. You can get strains of inbred mice that are all homozygous for genes that contribute to cancer. These are mice that are all guaranteed to develop skin cancer if you shave a patch and paint it with tobacco tar or something like that. You can get a very long time ago, I think we had someone working with, this would be 20 years ago, but we had someone here who I think was working with a strain of inbred mice that could be induced to develop these repetitive mouth motions that were considered to be a model for obsessive compulsive disorder in humans. So you can get strains of inbred mice that are homozygous for all kinds of genetic horrors. And I guess it's not a great deal of fun if you are one of those mice. But it does give you a system that you can use for, testing things like diabetes treatments or cancer treatments or obesity treatments. There are some strains of mice that are guaranteed to all get really fat if you feed them a certain diet. And you can study these conditions and develop treatments for them without dealing with genetic variation as a confounding variable. Now, you could create genetically identical individual mice by cloning, but that gets rotten expensive. This is a much cheaper way of developing strains of mice that don't vary, and also strains of mice that all have genes that model a particular disease. So anybody who goes on to biomedical research, maybe even some cell biology or neurobiology, even if you don't work with these yourselves, you will at some point come across research dealing with inbred mice. Yeah. And as Garen was saying, if it's 0, then basically there's no inbreeding. Inbreeding is a violation of the random mating condition. If the coefficient is 0, then mating is essentially random. The closer to 1 it gets, the more inbreeding there is. You actually can have inbreeding coefficients less than 0. They can be negative, which means that individuals are deliberately mating with individuals that are not like them. That's called dissortative mating. Okay, so yeah, F values are completely on a continuum. If F is greater than 0, it means there is some degree of inbreeding. And yeah, the greater it gets, the more inbreeding is present. Okay, that's actually a pretty good place to stop. So I'm going to go ahead and stop there and go ahead and stop the recording.